Hi, good evening, Year 13, and welcome to my next instalment and a somewhat emotional moment because this is your last ever key assessment. So, this is my attempt at Topic 8, Key Assessment 3. So, we start off with a nice multiple choice question. We can see that the question is about earthworms. So we start thinking straight away whether this is anything to do with obviously living things, working with living things. And then we look at this withdrawal response. So we're starting to think about potentially a little core practical that you've just done. Which of the following describes a change in the response as a result of repeated stimulation? So again, with these things, if you know the answer, great. If not, you've got to start to delete things that you don't think are going to be the case. Now, we know that reduction is all about things like photosynthesis, respiration. All right, inhibition, you would have come across inhibition, enzymes maybe. So you're left with two things, habituation and coordination. Coordination is a very, very general term for everything really to do with nerves and hormones. So that seemed a bit too generic for us. And the answer is habituation. Habituation, this idea that you can change your behaviour if you're constantly stimulated to make that change in behaviour worth your while. Okay, contraction of muscles and the withdrawal response is stimulated by nerve impulses. These nerve impulses can be detected using electrodes. Explain. So here we go. I need to know some biology here. The electrical changes in an axon that allow these nerve impulses to be detected. Electrical changes. Now, as soon as we're thinking about electrical changes, we need to be thinking about maybe some sort of potential difference, okay, i.e. a change in charge. Yeah, I'm sure physicists might disagree with me, but that's kind of how I think about it. All right, and actually, I'd be starting off by saying, well, if you're going to measure any change, there's got to be some sort of potential difference going on across the axon, okay? Because if this is our axon, we're going to get a change in different charge between the outside here and inside the axon, yeah? So we need to have a change in charge. So now we're thinking, well, we've got to explain, so we've got some biology in there. How's this actually coming about? Well, we know that the first thing that happens is that you're going to get the sodium channels, the voltage dependent or the voltage gated channels opening, and you're going to get the influx there of sodium ions. Okay, so as the sodium ion channels open, all right, you're going to get an influx of sodium. So Na open, therefore Na plus into the axon, okay? And that causes depolarization. Oh, can't spell, but you get the idea, okay? Now, once you've got the influx of positive ions going on in there, all right, the potassium ion channels then open in response to that. So the K plus channels then open, okay? And this time, because we've now got a lot of positivity, a positive charge within the axon, you're going to get the K plus ions going outwards. All right, so therefore, potassium ions, okay, are going to move out, and therefore you're going to get repolarization. I'll spell it right this time. So repolarization of the membrane. All right, not necessarily the easiest question in the world, but as long as you know that nerve impulse and you can relate it to this, then it actually becomes a bit more straightforward. Okay, as soon as I'm looking at electrical changes, I'm thinking potential difference. There's one marking point. How does this come about? Well, I know that my electrical impulse starts with the sodium channels opening, and therefore you're going to get Na into the axon or depolarization. In response to that, the potassium channels open, and therefore I'm going to get potassium moving out or repolarization of that membrane. So there's my 4 marks there. Okay. Right, one of these lovely 6-mark questions next. Okay, lots of reading for you to do there, so I'll let you have a quick look at that. Studies were carried out to investigate the withdrawal responses in earthworms. The duration of the withdrawal response is the length of time that the muscles remain contracted. If the stimulus is repeated, the withdrawal responses in each reduced in duration or lost. In one study, an earthworm was touched 20 times in 1 minute at point V, and the shortening earthworm body was measured after 20 stimuli, so it's only measured after 20. The effect of touching at point W in the same way was recorded, and also the effect of alternating touches between V and W. So the same way, so that's going to be relating to that. The results of the study are shown below in the table. Okay. In another study, sensory neurons of earthworms were stimulated by an electrode 6 times. They were recorded at positions X and Y. Nerve impulses recording at X and Y, shown in the diagram below. The presence of a line indicates an impulse was detected. S shows the stimulus. So I've got 2 pieces of information here. 1, 2. I know that I need to talk about 1 and I need to talk about 2. Now, I'm not going to keep switching over to the other side to write this as an answer. Okay, I'll do some annotations on here and you can see how I came up with my answer. Okay, so I'm going to talk about this first. What does this show? Well, this relatively straightforwardly shows that if you touch it at one point, i.e. at V or at W, then you habituate the animal. You habituate the earthworm and the body length doesn't change. Okay? If you stimulate it alternatively at 2 points, then you do get habituation. Sorry, you don't get habituation. The body length does change and the earthworm continues to react to the touching. Okay, so no habituation has happened. Why is that? Well, here you are touching at 2 places, so you're touching different nerves. Okay? You're touching different nerves, so you're unable to habituate that animal. Now, the second thing, all right, what's going on in the second one is that you need to look at this graph. So if I stimulate, then at X and Y, stimulate X and Y, stimulate X and Y, stimulate X, oh, I've lost Y. Stimulate X, I've lost Y still, and stimulate X, Y, so. So here, at this point here, I've got habituation. All right, but I've only got habituation at Y because I'm losing the Y's, but I'm keeping the X's. So you're going to lose Y after 3 stimulations, 1, 2, 3, okay? But you're always going to keep X. And now, because we're going to be explaining this, I think, analyse the data of these 2 states to evaluate, which it has some explaining in it, okay? Then you're going to have to start to think about, right, why X and why not X, and why does it habituate at Y? Well, there's a couple of things here. X is very close to the stimulation. The stimulation is happening here, okay? So it's doing that, so you're going to get a detection of that each time, but the other key thing here is the synapse. So we know from our biological knowledge that habituation is about synapses. So X is before the synapse, so it's always going to get there. The synapse is what causes habituation. So anything after the synapse can be habituated. So that's why Y is going to be a problem. Okay, so Y can habituate, but X can't. So that's what I've said about my 2 pieces of data. Now, when I evaluate, I need to come up with reasons why as well, okay? So I need to bring in some biological knowledge here. So I'm going to leave this table alone for my biological bit. I've written all my table data bits here, okay? And I've then got to talk about, well, what